Hello, dear student. Myself, Dr.Viral Shukla, Assistant Professor and Head Department of Microbiology, LG Institute of Applied Sciences. Today, we will complete with the remaining portion of translation. In translation part 1, I have clarified you with the role of ribosome and tRNA as well as the structure of tRNA. I hope you have gone through it, and today we will complete with the translation process. So let's move in detail about the process. So translation is actually a decoding of mRNA as well as covalent bonding of amino acid for the formation of polypeptide. Now this whole process is known as translation, and this particular process is going to occur at ribosome. So generally in bacterial cell, the protein synthesis rate is specifically the example is of Escherichia coli. It is having protein synthesis rate of around 900 amino acid per minute. So let's start with the understanding of this process. So this particular process contains three different stages: initiation, elongation, and termination. Now during initiation, the active tRNA, that is amino acid, it is attached with the tRNA. It's going to take a position on ribosome, and they are going to form an initiation complex, and that is known as initiation. During elongation, the amino acid addition will be there, and on a different side, that is known as EPA. And this particular process is known as elongation. During termination, that is a final step of the translation process, and this particular process takes place when the stop codon, that is UAA, UAG, and UGA, is come across to the ribosome. And during the termination process, the polypeptide sequence is going to be released into the cell. So let's move on. Now, before starting the initiation and elongation and termination, I just want to clarify the first important step of the translation process is amino acid activation. Amino acid activation means amino acid attachment to the tRNA. This particular process takes place when or catalyzed by amino acyl tRNA synthetase. This enzyme is very much specific for the specific amino acid. Means in bacterial cell, around 20 amino acyl tRNA synthetase is present, because each and every enzyme is specific towards amino acid. And during amino acid activation, one ATP is going to utilize. ATP is hydrolyzed, converted into AMP. Now, why this high energy is required during the amino acid activation? Because this ATP hydrolyzation is going to create a high energy bond between this. Now, this high energy bond is required during the polypeptide synthesis. That's why this ATP is hydrolyzed and converted into AMP, and this amino acid is going to attach at the 3' end of tRNA. Now, let's move on. Initiation. So, in order for translation to start, we need a few key ingredients. They include ribosome. They are in two pieces, that is 30th subunit and 50th subunit. They are the two pieces, large and small. mRNA with instruction for the protein will build an initiated tRNA carrying a first amino acid in the protein, which is almost always methionine. Now, here I want to clear a very important thing, that is in bacterial cell, modified activated tRNA is there, and that is known as FMAT tRNA. FMAT tRNA means formyl methionine tRNA. Now, in bacterial cell, the formyl group is actually known as an initiator, and it is going to hydrolytically remove during the process. Now, during the initiation, only methionine tRNA is going to employed on the site, not FMAT. Formyl group is going to be hydrolytically removed. But in bacterial cell, it is FMAT tRNA. Now, during its initiation, this species must come together in just the right way. Together, they form an initiation complex. The molecular setup needed to start making a new protein. So let's move on how this initiation complex is going to be takes place. Now see, when this FMAT tRNA, that is formal methionine tRNA, that is known as initiated tRNA, is going to attach with the AUG, that is a start codon. This AUG is just after the Shine-Dalgarno sequence, that is also known as ladder sequence. I already explained you during transcription and translation part 1 also, that Shine-Dalgarno sequence, that is a ladder sequence, it is not going to be translated, only transcribed. Now this particular sequence is after that start codon is there, and this initiated tRNA, FMAT tRNA, because AUG is coding for methionine. So FMAT tRNA is going to take place over here, and this is a 30th subunit, right? So here this is a start codon, this is a Stein-Dalgarno sequence, and this is a small ribosomal subunit. Now this complex is going to attach with the large ribosomal subunit, and large ribosomal subunit will come with the transformational functional groups, that is exit site and amino acyl site. So now for the formation of this initiation complex you have already seen over here, this is known as initiation complex. For the formation of this initiation complex, initiation factor role is very much important. So we will start with the role of initiation factors. Initiation factor 3, which prevents the 30s ribosomal subunit to bind with 50s up till the specific time. The role of initiation factor 2 is to bind with GTP, and role of initiation factor 1 is to remove or to give a signal to initiation factor 3 to leave 30th subunit for the formation of initiation complex. So let's move on. See, this is a IF2, which is coming with the GTP, IF3, which was actually attached with the 30th subunit, and now initiated tRNA will come over here, IF3 will leave, so 30th ribosomal subunit will be free, and this FMET tRNA, initiated tRNA with initiation factor 2 will come over here, and IF1 will give a signal to leave IF3. Now, as IF3 will release, the 50th subunit will come over here, and GTP, which has come with initiation factor 2, will going to be converted in GDP. So GTP is converted to GDP, and this 50th subunit is going to form a 70S initiation complex. When 50S has not entered this complex with active tRNA and GTP is known as 30S and mRNA, this is known as 30S initiation complex. And when this particular 50S subunit comes together, means 50S subunit, 30S subunit, mRNA, and FMAT tRNA, this particular complex is known as 70S initiation complex. Okay, so once initiation complex forms, now the most important step, and that is a second step of translation process, and that is elongation. Elongation actually divided in 3 phases. The first phase, that is known as amino acyl tRNA binding. Second phase, that is known as transpeptidation, and the third phase, that is known as translocation. So let's move on how these phases are divided. Now the FMAT tRNA start out in the middle slot of the ribosome, means the FMAT tRNA, that is modified tRNA, which is present into the bacterial cell, formal methionine tRNA, always takes place at a P site. So the initiation always takes place on P site. Now the next active tRNA will takes place at A site. So first P site and then A site. So a fresh codon is exposed in another slot, and that is known as A site. The A site will be the landing site for the next tRNA, one whose anticodon is perfect, means complementary to the anticodons. And that is known as amino-acyl tRNA binding. The next step, that is known as transpeptidation, means the peptide bond is going to be formed during this step. This step transfer the methionine from the first tRNA onto the amino acid of the second tRNA in the A site. So P site amino acid will be shifted on A site. And this particular combination will happen between the N terminus of the methionine, means from the P site N terminus, and from the A site C terminus, means amino group will going to be contact with the carboxyl group. So the methionine forms N terminus of the polypeptide, and other amino acid is that C terminus, that is known as transpeptidation. Now, once the peptide bond is formed, the mRNA is pulled onward through the ribosome by exactly one codon. This shift allows the first empty tRNA to drift out via the E site. So, from A site, tRNA will move towards, from the P site, it also exposes a new codon in A site, so whole cycle can repeat. So, during translocation, peptidyl tRNA moves from the A site to the P site, and ribosome moves one codon along mRNA, and new codon will take position. And the empty tRNA moves from the P site to the E site. That is known as translocation. So let's see by the figure, we'll understand about that. Now, in this translation process, the most important factor is elongation factor. So as you can see from the figure that elongation factor Tu and elongation factor Ts is involved in this process. Elongation factor Ts is responsible for activation of elongation factor Tu, and when activation will be there, elongation factor Tu is going to attach with the GTP. Now this elongation factor Tu GTP complex is going to attach with the tRNA, and this EFTU GTP is going to attach with the tRNA. They are going to form EFTU GTP amino-acyl tRNA complex, and they are going to attach with the A position. And here GTP will be utilized, converted into GDP, and from A site, it is going to, this polypeptide chain is going to move on P site. So now this elongation factor, EFTU and EFTS, I hope the role of both are clear to you. Now, the last process, that is a termination process. Now, in this termination process, before starting termination, I just want to clarify one more thing over here, that in final phase of elongation, three step will simultaneously takes place. First one, that is peptidyl tRNA moves from A site to P site. Second, ribosome moves one codon along with mRNA to take a position on new codon. And the third step, the empty tRNA moves from P site to E site. This three process in a final phase, that is known as translocation, will take simultaneously. So this particular process, you keep it in your mind, because elongation is very, very elaborated process into this translation cycle. Now let's move on the termination. Now termination is the stage in which the finished polypeptide chain is released. Now this process is also known as when protein synthesis ends, when ribosome reaches a stop codon, one of three stop codon, that is UAA, UAG and UGA. Now this particular codon are not actually recognized by the ribosome itself, but here release factors are involved in the termination process. Three release factors are there, RF1, 2 and 3. They are going to aid the ribosome in recognizing these codons, and they are going to hydrolyze the bond by the use of enzyme peptidyl transferase, and they are going to linking the polypeptide to the tRNA in the P site, and the polypeptide and the tRNA are released. Empty tRNA are released from the uh, ribosome, and one GTP is going to be hydrolyzed during the process. So let's move on the detailed process. See, RF1, RF2 and RF3 are involved in this termination process, and GTP is going to be hydrolyzed during the process. One more, elongation factor G is also involved in termination process. So let's see in detail how it takes place. So see, RF1 is going to bind, and it is going to take place at the position of A site. Here is going to take place at A site. Then RF RF3 is going to bind with this, and GTP hydrolysis will be there, and here GTP will be converted into GDP, and from here, when GTP is going to be hydrolyzed, RF1 and 2 factor will release, and RF3 is also going to release. Now when EFG is entered, once again the release factor is going to bind with this process GTP EFG, and they are going to bind at A position, and GDP RRF and EFG complex will release. 50S subunit will be dissociated, and its initiation factor 3 is going to attach with this ribosome, and now tRNA is going to leave the position, and now this 30S subunit is attached with IF3, so it is going to stop, or it is going to initiation factor 3 is having a role to put aside a 30S subunit, or to keep it, or to stop binding it with 50S ribosomal subunit. So now once again, this is a translation cycle. What I have explained you, that elongation factors, initiation factors, and release factors are involved in whole cycle. And let's see about the energy requirement during the whole translation cycle. 2 ATP is required for the activation of amino acid. 1 GTP is required during the initiation of translation. 2 GTP is required during the elongation of translation cycle, and 1 GTP is required during the termination cycle. So once again, 2 ATP and 4 GTP is utilized during the whole process. So I hope you clear with the translation today. My reference was Prescott's Microbiology 10th edition. Thank you everyone. Have a nice day.